Well, while people are queuing up, so is it fair to say for PKD that given that there really aren't any new options, that maybe more awareness of the incidence of PKD may be worthwhile? Is it something like, getting some sort of volume imaging of children at 10 and then at 20 to be able to recognize whether or not there's been an increase in volume prior to seeing any loss of kidney function?
Arlene Chapman at the University of Chicago thinks based on ultrasound imaging and MRI imaging, she can predict the severity of the course of the disease in a rough sense. But there's also the question, and I know this because of my own children, as to whether you really want to tell a 10-year-old or a 15-year-old that you may be diagnosed with a disease with no treatment. So that's a philosophical question. I mean, I tell my kids, avoid caffeine, drink a lot of water, watch your diet. Frankly, things that anyone who's interested in their health should do.
So I have a question, Jim Beck from the Parkinson's Foundation, for Dr. Humphreys and Dr. Gompertz, because what was interesting is we're talking about, most of the talk is about cellular strategies that involve maybe single cells that can then propagate, but we're talking about three-dimensional structures. And so I was wondering if you had any insight about acellular structures or maybe 3D printing with some kind of biomaterial as ways with which to kind of get over this hump of where you need to really create a three-dimensional structure to have a functioning organ.
Yeah, that's a great question, and that is an area of interest within the field, certainly in kidney, I think also in lung, where what folks have done is taken a kidney and used gentle detergents and bathed it in these gentle detergents to create a decellularized scaffold, which is basically all matrix proteins, which have important niche cues for the cells that used to live there. And then they've reinfused progenitor populations, not always well-characterized progenitor populations, and there's some data that the cells do tend to differentiate in some ways that they should, but in other ways that really, where the cells are in places that they shouldn't normally be. My personal view, I think it's an important area, and we need to hedge our bets and not rule anything out, because as you point out, these are such complicated organs that it's hard to predict what will bear the most fruit. But this scaffold approach to me is sort of just really sort of like forcing something that's a little bit unnatural, whereas what I like about the directed differentiation approach is that when you get these populations of progenitors that are present in development but not in the adult, the ureteric bud and the metanephric mesenchyme, when you combine them in a dish, they simply do what they want to do, which is create nephrons. And right now they can create 200 in this little tiny ball. But to me, it's just a question of scale and time and conditions to change that to 2,000, to 20,000, and ultimately to the 200,000 to million nephrons that are present in an adult kidney. So I think it's important to pursue that avenue, but it's not where I would put my money. I don't know if Brigitte would agree or not.
So Ben, I would also say that a spin-off of investing in kidney organoids is not just what you get at the end as something which might be therapeutic, but also as a research tool and an assay tool, right?
Yeah, I'll just say briefly that if you look at cystic fibrosis and what Aurora Biosciences did more than 11 years ago, was they had an in vitro screen for the chloride channel, and they really didn't rely on an animal model at all. So high throughput screening was really effective. However, in PKD, and I'll let Dr. Humphreys speak to the other nephropathies, there has been no in vitro model for high throughput screening, and one has to rely on various rodent models. There's actually Persian cats have a mutation. English bulldogs have the equivalent of ADPKD, but these are not species that one normally uses for research. Yeah, herding cats is, or dosing them is, and honestly, the FDA would have an issue with a new non-rodent species like the cat. There would be a lot of regulatory hurdles there.
Theodore Erna from Altius. I have a question for Dr. Barron. Thank you for a very thought-inspiring presentation. You know, in the gene therapy and the gene editing space, we broadly speaking can approach a gene addition or a gene knockout strategy. And it seems that PKD, while autosomal dominant, has to do with haploinsufficiency of PKD1. In other words, we have to put it back in, and as you mentioned, it's too large for most traditional delivery vehicles. In the sickle cell disease and beta thal space, there has been really remarkable progress looking at genetic second site suppressors. So Stu Orkin at Harvard identified this gene called BCL11A, the loss of the expression of which is disease protective for individuals with normally sickle and thal causing mutations. And that got us all in the gene editing and gene therapy field excited, because this means we can knock a gene out and complement the mutation in beta globin. I was really struck by your point that some people with mutations in PKD1 or 2 don't really get severe disease. And I'm wondering if that could potentially be also due to second site suppressors. Have people looked genome-wide whether there are extragenic suppressors of mutations in PKD1 or 2? Because if there are, those could potentially represent druggable targets.
I wish I knew the answer, and there may be an answer to your question. Definitely haploinsufficiency is considered the second hit, but many researchers in the PKD space are considering possibly third hits, that haploinsufficiency alone may not explain the disease. And obviously environmental factors determine how many nephrons. Say you have 1 million nephrons, probably 1,000 or 2,000 are actually forming cysts, and what that stochastic probability of a given nephron forming cysts is, and why they become haploinsufficient is unknown. But I think your comments are certainly worth investigating, and I wouldn't be surprised if those in the modifier arena are looking at that.
Is there any direct, similar to what Deepak talked about in cardiovascular, are there any targets in the kidney itself for trying to, I know it's so complicated in terms of what the chicken and what the egg is, in terms of which cells are dying first, but is there any approaches to trying to regrow glomeruli in situ?
Well, so,
Self-aggregates,
yeah.
They do it in vivo.
Right, so there are species which can grow new nephrons, and fish primarily. And these fish, over the course of their lifetime, as they get bigger and bigger, they deal with the need to basically filter a larger blood volume by sprouting new nephrons that sprout off of the collecting duct, and they're really fully functional. And it's always been a mystery and about why that ability has been lost in mammals and in humans. And it has been something that's been investigated really with zero signs of success to date. But the notion that you could somehow reactivate developmental signaling pathways in the right cell type, whether it's mesenchymal cells potentially or others, is a super attractive possibility. Obviously, you'd need to balance carcinogenesis with proper development, but I think the fact that fish can do it, gives one at least the hope that somehow those pathways might be reactivated in mammals.
Any other questions? We might
Perfectly on time.
almost
Oh
be
my God.
back on time. Yes.
Yeah, I think all
Thank you, Ben and David.
Thank you